Okay, let's get started. My name is Richard Kondo. I am VP of Strategic Partnerships at AnaBios, and I want to thank you for joining us today for another round in our webinar series on investigations of biomedical issues and how they relate to human disease. Can I have the next slide, please? First, I'd like to announce the Translational Research Grant. AnaBios is committed to enabling human-focused drug discovery in areas of high unmet medical need, including cardiac disease, chronic pain, and respiratory ailments. That's why we're pleased to announce the third annual AnaBios Translational Research Grant for a net award of $15,000 US. The $15,000 award of high-quality human tissue samples from AnaBios will help the winning researcher advance his or her translational research project. In addition, we will award a 25% discount to the 10 highest ranked applications for future purchase of AnaBios tissue samples. To submit your application, please upload your abstract and a detailed explanation for your use of human tissues at the website that appears on your screen here. The deadline to submit your abstract is July 1st of 2023. Award winners will be announced on July 30th. Please visit our website for terms and conditions. So we'll have a Q&A session at the end of the presentations. To ask questions, please type your question in the questions box that's shown in the webinar panel on the lower right. Questions can be answered during the webinar, but I will present the questions to the speakers after the finish of the presentations. So now on to our speakers. So next slide. Okay, thank you. So our first presenter is Dr. Jeffrey Skolnick. Dr. Skolnick is Regents Professor of Biology at Georgia Tech. His research has focused on the development of computational algorithms and their application to proteomes for the prediction of protein structure and function, and the prediction of small molecule ligand protein interactions with applications to drug discovery. One of Dr. Skolnick's key insights is his demonstration that the number of ligand binding pockets in proteins is small, providing scientific justification that large-scale drug repurposing will work. Dr. Skolnick graduated summa cum laude from Washington University in 1975 with a BA in chemistry, and he obtained his PhD in chemistry at Yale University. Our second speaker is Dr. Andrea Getty. Dr. Getty is Chief Executive Officer at AnaBios. A neuroscientist by training, he is focused on the creation of new tools and strategies to advance biomedical sciences, with special emphasis on enabling of the direct study of human biology and pharmacology to accelerate the rate of successful drug discovery and development. He was formerly president of Aviva Biosciences, where he led the growth of the company into drug discovery services and intellectual property commercialization. Prior to joining Aviva, Dr. Getty was involved in drug discovery programs at Aurora Biosciences, now Vertex Pharmaceuticals. And Dr. Getty received his PhD in molecular and cellular physiology from the University of Verona and did his postdoctoral training at Caltech. So thank you very much. The title of today's dual presentation is a novel approach to utilizing AI and biological assays to help address the opioid crisis. Dr. Skolnick.
Thank you very much. Good morning. So yes, so I'd like to begin by kind of giving a background as to it's kind of obvious to anybody who knows, but why focus on chronic pain? So if I could have the next slide, please. I mean, if you just look at the statistics, and this thumbnail sketch off to the left gives you kind of a feeling for what is the problem. I mean, over 225 million people daily suffer from chronic pain. A significant fraction of those report a lot of pain. And if you look at the statistics, and if anyone has ever suffered from chronic pain, it is really a life-changing and quality of life diminishing experience. And if you got to ask the question, so what is the state of the art? If you were to try to develop a anti-pain drug, and ideally what you want is a pain drug which is non-addictive, which is even a higher barrier. So it should be a non-opioid. It's got a 2% probability of success. So it's 5 times lower than the generic success rate across all diseases. And so the approach which we've adopted, and I'll have the next slide, please, is basically what we want to do is use AI to focus on what are the relevant regions of chemical space to do experiments on. In principle, if you had infinite resources and infinite time, you could screen every molecule known to man and every molecule that man might be able to make, and you might test it, and eventually you may come up with a perfect molecule. In the real world, it doesn't work that way. So what you want to basically do is use AI to do 2 things. Understand the underlying physical processes, which are quite complicated with a lot of feedback loops. It's kind of like juggling 1,000 balls at once to figure out what are the key drivers of a particular type of pain, then identify a set of molecules, hit ultimately leads that then gets optimized, which then get fed into the laboratory. The laboratory then tests it and says, yes, you did a great job. No, you didn't do a great job. Fix it for those which look like molecules, and then almost becomes a traditional drug discovery and lead optimization problem, except we're using a different set of tools than the standard of care to do this. If I could have the next slide, please. So basically, what we want to do is at the beginning of the pipeline, we want to do the heavy lifting from AI. If you had algorithms, which I will tell you about, which do give you a probability that they're going to work before you actually do them. And so what you want to do is identify novel targets for pain, and then screen them virtually. And then, as you've seen in the next slide, you pass it to the experimentalists. They say you buy them or you test them. You now have your library of first round molecules. And then in the next slide, what you do is you validate the subset using a biochemical assay. You then learn from your mistakes. We retrain the AI, a new round of hits are generated. You then go make the new molecules, iterate, repeat, and you keep on doing this until you have the desired clinical response and activity. You know, whatever concentration profile, bioavailability, all the standards of normal drug discovery that operate. So basically, the marriage of this is you want to use the AI to do the high throughput and the experiments to do low to medium throughput. And this is particularly relevant for things like if you're screening in dorsal root ganglia, where you can't screen 100 million molecules in 100 million dorsal root ganglia at the same time. So this actually enables it to look here, not there, and improve the efficiency of the algorithm and focus one's experimental efforts on productive regions of chemical space. If I can have the next slide, please. Okay, so what is the key idea behind this? I mean, one of the key ideas is one might imagine if you have a small molecule, it fits into a pocket, like in this nice little circle with this kind of very naive lock and key idea. Traditionally, when people assumed is that if you have a protein, you're going to have literally tens of thousands of distinct binding pockets. In reality, what turns out, and this covers more than 97% of the human exome, is there's actually on the order of 500 distinct stereochemically relevant ligand binding pockets. And this also explains both the good news and the bad news. If you have a pocket which is very similar, it explains one of the reasons why you have drug side effects and hit undesirable alternative targets, because the pocket space is actually quite small. And what's really remarkable is you will see stereochemically similar pockets, pockets with the same shape and the constellation of residues in the appropriate place in proteins that are an example of convergent evolution. So this is actually a stereochemical feature of the protein structures themselves. And of course, what you can do is you have a set of binding pockets, and you can use this for optimization, for example. So you have a pocket in the target protein you love. And then you can ask the question, if I look through the entire exome, what are the set of alternative pockets that look very similar to the pocket I want? If they're too similar, and this alternative protein has a miserable side effect, like let's say QT prolongation or death or something like that, it means that you picked the wrong pocket and there's no point in proceeding further. On the other hand, if the pockets for the alternative targets look like they're designable and the medicinal chemist or an AI algorithm in combination with a med chemist can actually optimize it, you can actually eliminate side effects. So if I can have the next slide, please. So basically, there's a set of features which you want. Okay, what you want to do is, and I'll just give you a very high level qualitative feature. Okay, the library of pockets is small. That's great. So what can you do? So basically what we want to do is just predict ligand binding sites and ligands that might potentially bind with a confidence index that they're going to bind at a 10 micromolar or better threshold. So what you do is you run out and you find a bunch of templates that could be structurally related or not. You compare the pockets in the templates to the pocket in the target protein of interest. Now, what is very nice is you can use both experimental structures and predicted structures. And this was even true before the advent of AlphaFold 2, which generates a much better set of models for many more proteins. You've now identified potential binding sites that have not been targeted by existing drugs. You find similar binding sites in different proteins. So this is the drug repurposing. You know, I have a similar stereochemical structure in another protein. I know the side effects of it. It's safe or not so safe, depending on the use. You can use this for repurposing. Once you identify a potential drug hit, and this is all small molecules, this is nutraceuticals, synthesized molecules, naturally occurring molecules, it really doesn't make a difference. You predict how modifications in the drug might influence its binding, because what it actually does is it looks at the subpockets and the stereochemical features of the subpockets. Some subpockets are remarkably promiscuous and are just kind of like generic Velcro. I mean, if you have a hydrophobic ball, for example, it's going to interact everywhere. It's not specific. But if you happen to have it in the neighborhood with other things that drive the specificity, you can use it to improve binding affinity. And so that's kind of at the high level what the virtual screening does. The second thing is we've developed a machine learning algorithm called efficacy, which is actually designed to predict the mode of action proteins for 3,608 indications at the moment it's been trained on. And what it basically does is it says, these are the set of proteins that are associated with this phenotypical response. So it's actually going from a single target approach to a collective target approach. And it basically uses as a form of AI called posted random forests to predict potential human targets. It can predict drug side effects, at least one crappy side effect, death, stroke, heart attack, cancer with greater than 80% success. It also predicts indications. So if you do this in screening retrospectively against all drugs and you pretend you don't know it, it can predict drug the efficacy over 80%. And what it does is it gives you, if it happens to work, it not only tells you, is it likely to work, but why? These are the players that are probably important in dictating why the drug works. So it can improve efficacy. And this is particularly important. And as an example, we're going to be showing you the case of applying this to predict molecules that have little, if any, known analgesic effects. And could we use it? So if I could have the next slide, please. So basically, this is kind of the protocol. How this was trained. We took a whole bunch of drugs that we trained with pain. We trained them on efficacy to find pain indications, the corresponding drugs. We then screened the entire FDA library of probe drugs to define mode of action proteins that are actually behind this. So this also looks at genetics. So it's actually a bit more complicated than this. We predict protein targets in the binding sites. You then do virtual screening. You iterate, repeat. And this is how the algorithm at a very high level works. So it's trying to develop. Now, what is also tucked in here, which is really important, is we identified, and this was actually due to my son Sam's Skolnick's idea, that there are certain molecules like ibuprofen. You may bleed to death and get a heart attack, but they're not addictive. And so what we actually asked the question is what is the mode of action of the pain drugs with pain from drugs without pain? And so what we actually do as part of this de-risking process, it turns out that the drugs that actually are non-addictive suppress addictive responses in addition to the analgesic effect. And so the molecules that we provided to AnaBios to screen were actually de-risked for the likelihood that they're addictive. So you get 2 for the price of 1. And again, these are probabilities. It's not perfect, and you got to test them experimentally. So if I could have the next slide, please. And so at this point we're going to have round 1 hits. You screen them in dorsal root ganglia, which Andre will be telling you about in a minute. These then provide scaffolds to screen an E-molecule library to find a bigger set of hits. These then are privileged structures for synthesis, and you keep on iterating and repeat. Operationally, we've completed steps 1 and 2. We have not yet synthesized the molecules, but that's on the plate of the to-do list. So we'll tell you, and Andre will now take it over and tell you about what happens when you apply this in the real world rather than the in silico world.
Yeah, thank you, Jeff. So as Jeff mentioned, in this collaboration between Georgia Tech and AnaBios took care of the biology side, and we have focused in particular on the activity of the compounds identified by the AI on human sensory ganglia. Why did we select sensory ganglia? So these cells are critical components of the pain pathway and provide a very promising target for the development of non-addictive analgesics. Peripheral sensory neurons are located outside of the central nervous system and are responsible for transmitting pain signals from peripheral tissues to the spinal cord and brain. In targeting these neurons, it is possible to block or modulate pain signals at the source before it reaches the CNS. There's another important advantage of targeting peripheral neurons, and that is that it's basically minimizing the possibility of having addiction-related side effects. Now, another important feature of these cells is that they express a variety of molecular targets, such as ion channels, G-protein coupled receptors, and other signaling molecules that are known to contribute to pain sensation. So how did we obtain human sensory ganglia for conducting the in vitro test? So AnaBios specializes in the recovery and interrogation of a variety of human tissues and human cells. We do this by leveraging a network of hospitals and transplant centers across the country that collaborate with us. And by employing proprietary tissue recovery methods and solutions and advanced shipping logistics, we're able to recover a large number of human organs and tissues on a weekly basis. So all of these samples are received at our facilities, either in San Diego or the facility in Philadelphia, and are used for either biobanking or for live tissue or live cell type of assays. And some example of the cell type and tissues that we received are shown here. So at the top left, for example, you see contracting cardiomyocytes isolating from all human hearts. We also have the ability to look at disease hearts and conduct studies, for example, on atrial fibrillation or heart failure. We work on lung tissue and lung slices. And down at the bottom, you actually see 3 different tissues that are relevant to the pain discussions we're having today. So on the left side are recording from dorsal root ganglia. These are electrophysiologically measured action potentials. In the center is calcium imaging on a human spinal cord slice, and on the right side, these are again recording from trigeminal ganglia. Now, typically the dorsal root ganglia we recover from the thoracic level all the way down to the lumbar level, and the ganglia are dissociated upon arrival in our laboratory. They are dissociated to establish primary mixed cultures of neuronal and glial cells. And these cultures have exhibited good phenotypic stability for at least a couple of weeks. And this gives us enough time to conduct both electrophysiology-based measurement as well as calcium imaging. For each case, we have good annotation regarding the donor's health status, gender, or any kind of medications that they were taking. And what is interesting is that we have access to both normal dorsal root ganglia as well as ganglia from donors that were affected by one or another form of chronic pain. So there is the opportunity to test compounds not just in the normal phenotypic background, but also pathological states. Now, specifically to the collaborative project with Georgia Tech, what we have decided to do is to employ an assay which specifically tests the activity of compounds in the context of dorsal root ganglia that have been sensitized with inflammatory agents. So typically, nociceptive neurons do not fire action potentials, are not activated unless there is a peripheral noxious, painful stimulus. However, in the context of inflammatory sensitization, there is a significant amount of spontaneous activity in nociceptive neurons. That means that even in the absence of a noxious and painful stimulus, the cells can fire spontaneously action potentials. And this is the assay, the foundation of the assays that we have utilized. So we have in calcium imaging, we look at spontaneous activity under baseline condition in the absence of any kind of stimulus. Then we treat the cells with inflammatory sensitizer, and what we see is that the level of spontaneous activity is greatly increased, as you can see in the bar graph at the center of the slides. And then one can test a number of drugs or articles for the ability to inhibit the spontaneous activity initiated by inflammatory agents. This is a fluorescence calcium imaging assay in live neurons that is conducted at physiological temperature and is typically run in 96 well plates with the throughput that can be as high as 150 molecules per day at a single concentration. And this assay, in the project so far has been conducted using normal DRGs, but as I mentioned, it could be extended to pain donors to further validate the hits. And some examples of the results we have obtained. So on the left side is a raster plot showing the baseline control in which, as you can see, there is a very low level of spontaneous activity, increased spontaneous activity in the presence of prostaglandin and bradykinin, and then the effect of, in this case, local anesthetic sodium channel blocker lidocaine that can drastically reduce the level of activity in the neurons. And some of the hits from the screening that we have conducted on the molecules identified by the AI provided by Georgia Tech are shown on the right side. And so you can see that one compound in particular, fingolimod, exhibits a level of activity which is comparable to that of lidocaine. So this is an example to illustrate the concept. Obviously, this particular assay format is not the only one that can be used. One can bias differently the selection of compounds. For example, instead of using inflammatory sensitized dorsal root ganglion neurons, one could use electrical field stimulation to specifically concentrate on compounds that are active on voltage-gated ion channels. But this is just one example of implementation of this concept. And now I'm going to give the presentation back to Jeff to discuss further the results of the project.
Thank you, Andre. So if we go to the next slide, basically 11 molecules were tested, and by the criteria of the assay and statistical significance, 4 of 11 at 36% were initially successful, and those are the little molecules sticking off to the left. Fluvoxamine, ibutylide, cyclopentanate, and fingolimod were the first. So it's not a bad success rate. This is a phenotypical response. I just want to point out, this is not just simply binding to a molecule and you say, yes, I hit the target that I wanted to hit. this is, at least with respect to the DRGs, a phenotypical outcome, which isn't bad, considering all we started with was the chemical structure and a bunch of proteins, and no other extrinsic information was provided. If I could have the next slide. So this is kind of, showing, the cluster centroids of, we then use these as seeds for round 2, and this gives a collection of molecules which may be interesting. We excluded molecules whose tiny motor coefficient, the measure of chemical similarity, is greater than 0.8 to the inactives. And these are kind of some of the top ones that come up in the algorithm. And what it's also showing is if you look in column 3, this is the predictive precision. What is the probability that you're going to see a phenotypical response? And it's kind of saying it's on the order of 8 to 10. So those are the kind of numbers we like to use to feed to the experimentalists, so experimentalists don't waste their time looking in the unproductive, likely unproductive regions of chemical space. If I could have the next slide. The next part of this process, which I'll move to, was can we then use this, because this was an NCATS project, to actually do automated synthesis. And so we fed some optimized molecules to the ESCP portal of Nicole Young and her research group, and they, basically feed it into their engine, and they give synthetic steps. So you could tie this to automatic synthesis tools and protocols if you chose to. This is a proof of principle to do that. If I can have the next slide. And this is just showing, fluvoxamine, which is a second round novel analgesic. It's, it's not as good as lidocaine, but at least it kind of, in this baseline experiment, reduced it for that case. It kind of worked. So, this is just an example. And again, we're screening handfuls of molecules for anti-pain response. And so, we were convinced that this is a promising first pass study, and it has it's a useful tool for a combination of molecular prioritization using AI, which also gives you some potential insight into what are the potential pathways that you're tickling, as well as relevant hits in which to test in the laboratory. And so, if I might have the final slide, please. So basically, this is kind of a proof of principle study of a comprehensive platform for translational innovation in pain, opioid abuse disorder, and overdose. And again, what it does is we're going to use AI-based tools as prioritization of targets, modes of action with human tissue to actually validate the hits. And then the biological data and the structure-activity relationships from the previous round are used to refine the AI so it looks for molecules in a certain region of chemical space. And this is iterative. And hopefully, if you keep on doing it, one of the things which I do want to point out is we discovered new chemical matter in round 2. This was not simply incremental SAR, where you're doing the methyl ethyl propyl series, which of course is all reasonable. It actually looks at the key functional groups of the ligands that are binding and putatively involved in the function, and can skip into different regions of chemical scaffold space. And the thing I want to leave you with is that a key feature of this approach is it appears to predict small molecules, which are, at least in retrospective studies, are likely to be non-addictive. And so it further de-risks the drug discovery process, because the worst thing is nobody wants to come up with another molecule as effective as fentanyl and even more addictive. So that's basically what we have done. And this is kind of an interim report. There's clearly much more work in studies that need to be done. But Andre, AnaBios, and our group are confident that this is a promising study that can help accelerate the discovery of novel molecules with anti-pain properties. So I thank you, and thank you for showing up for the webinar, and we'll be happy to answer any questions, Andre and I, that you might have.
Thank you, Dr. Skolnick and Dr. Getty. So as I mentioned earlier, there is a questions panel. That's in the control panel. I hope you see it. And you can type in your questions. I will read the questions out for answering. So while we I have a question while we wait for incoming questions. So I guess this is for you, Dr. Skolnick, which is to understand again the process by which the computational algorithms work. You are looking for ligands that have binding pockets, bind to binding pockets in proteins. In this particular project, of proteins that are known to be expressed in sensory neurons. Is that the universe that you were looking at?
So the first thing, because obviously, is you could say, I'm going to predict you're going to bind to this magnificent protein, and it's not expressed. So the data is actually filtered. What we have is a set of putative mode of action targets for the particular indication. In this case, pain, though it need not be. And then those mode of action targets are further filtered for protein expression in the relevant tissue. And so we look at the subset that are predicted to be interesting that are also present in the tissue. And then we do the virtual screening against this library of potentially, targets where we want to inhibit pain. So that's how it works.
Okay, thank you. And what about all of the compounds that are already known? You're looking for unknowns, or repurposed unknowns. What about all of the known analgesics that work on sensory neurons? Were they deliberately excluded? How does it work with that?
So the answer is it depends on the question you're asking. So in the training of the algorithm and the validation that it has value, what we typically do is we cluster them at tiny motor coefficient to 0.8. So I don't exclude one molecule that differs by a marginal change in my training set to figure out which are the pain proteins. And then we have 10 to 20% of proteins that are, small molecules that are orthogonal. We feed them into a library and say, okay, this is very nice. Of these 10 to 20%, which is on the order of like 15 to 20 distinct molecules, how many of them are predicted to have analgesic effects? And that's one way of validating the algorithm, and the answer is about 85%. So it's not perfect. But then you do this by retraining the algorithm, and you basically ultimately work through all the known anti-pain molecules, but there's a well-separated training and testing set, so it's not memorizing. And then the average performance is about 85% for identifying analgesic molecules effectively. So that's how that's done.
Okay, thank you. Well, I want to thank both our speakers for a very interesting talk. I will note that for those who would like to listen to the talk again, or for those, we will be sending out an email with the recording of the webinar, a link to the recording of the webinar to allow you to listen again, and for those who were unable to make this particular recording of it. But again, I want to thank Dr. Skolnick and Dr. Getty for the webinar, and I hope you have a great day. Thank you.